Okay, hello everyone. I hope wherever you are, at what time of day, it is probably early morning like in Canada, midday or late at night in New Zealand. I hope you all had a good day and great time. It is 10 p.m. in Sydney time. And I think I'll need to, yep, turn it off. And you are hearing me from Northern Australia. My name is Ali, and I work at the CDU as lecturer in Hematology and Transfusion Science. And I'm actually committee member of the International Society for Advancement of Cytometry. It's a great pleasure to be with you guys, and it's quite exciting to be part of a global hematology scientist. And I'm myself learning a lot from this group. Okay, let's get started. Today we are going to talk about the flow cytometry. All right, one more attendance. Okay, right, here. So, what is a flow cytometry? So, what is a flow cytometer? One of the questions which often used to be asked about the flow cytometry and the difference between a flow cytometry and automatic or auto analyzers was that. So basically, if you look at the printout of almost all of the auto analyzers, you can get a printout in a few seconds. And as long as all the quality control measurements are in place, you can trust the results. Whereas if you want to get a printout of the cytometer result, you'll probably get something like this. Yes, that's pretty much nothing. So that is more subjective instrument. So the reason I put it like that, I wanted to say that this is a subjective instrument. So from the very earliest steps, from sample preparation to something happened, right, to instrument setup, and all the way down to the data analysis, at any of these steps, if something goes wrong, still you will have a result, but the result is not reliable and you cannot trust it. So that's why it's a subjective instrument, and the same sample may be analyzed by different people, but at the end, as I said, at the data analysis, you still may get different results. So let's see how this system actually works. So yes, first, because in flow cytometry, we take advantage of the fluorescence technologies. I'll very briefly go through the fluorescence technology, that how the fluorescence technology actually works. So fluorescent material are compounds that once they cut the energy and they get excited, the electrons jumped up to a higher level, and once the electron is turning back to the ground state because it is unstable, then the extra energy will be released in the form of light, which is called fluorescent light. So this emitted light is usually, or let's say most of the time, it is greater or longer wavelength. So fluorescent material are excited with a particular wavelength of light and then emit a greater wavelength of light. And obviously they could be in different colors. They could be green, they could be red, they could be yellow, or etc., etc. And once we couple these fluorescent material to antibodies, then we have immunofluorescence. Right? And you know that now this is a very common technique which is used in different methodologies. So basically, if we have a cell with different antigens and we have a conjugated antibodies which is targeted against any of these antigens, we can detect these cells under the microscope or any other system which can illuminate the fluorescent signals. And in this case, for example, we have two antibodies which are conjugated with two different chromosomes, one red, the other one is green, so we can have a multicolor identification. And this is called direct immunofluorescence. And if we want to have a greater signal, right, then we can use the indirect immunofluorescence, which is basically we use a primary antibody against the target, and then we identify that with the second antibody. So basically, because for each of these primary antibodies, there are a couple of these secondary antibodies, then we have a greater signal. So each time that I accept someone in, then yeah, that was my. Maybe I should. Yeah. Okay, so this is indirect immunofluorescence. Yep. And I'm sure that you have seen this sort of immunofluorescence pictures which we use in the microscopy, immunofluorescence microscopy. So as you can see, for example, in this picture, we have 1, 2, 3 colors, one dappled which stain the nucleus part and the side parts with different colors. So you may say, what's the difference? So why we need the flow cytometer then? So how many cells can you count on this on the microscope? Not many, but the art of flow cytometry is that you can analyze 100,000 cells in a few seconds. And not only the analyzing, but also you can physically sort those cells. So now let's move on to the flow cytometry technology. So flow cytometry basically is made of 3 basic parts: fluidics, optics, and electronics. So fluidics part is the place where allows us to have single cells to be detected and analyzed, and then we have optic parts which collect the lights, and then the computer to analyze them. Okay, so let's go to the fluidics. So this is a very sophisticated engineering stuff, so I'm not going to go very deep into this, but just touching on the basis. So fluidics is made the technology which is called hydrodynamic focusing. So basically we have a 2 stream of fluids which are under 2 different pressure and running through the system. And because these 2 fluid streams are not in the same pressure, so they're not intermixing. So the sheath fluid sort of as like a sheath surrounds the internal or the central stream, so that once we have a sample injected in the middle of this central stream, then the sheath fluid sort of align the cells to just travel singly. So this single file through let the cells or any particle to all the way travel through and go to the waste. So this part of the cytometer is actually called flow cell, and it has a very special properties, especially in terms of the light scattering. So once the cell passing through this pathway, they can meet the source of light, and they can pass through this source of light, which in most of the cases is actually a laser light. So here is the place that cells can scatter lights. So, for example, some of the lights can pass through the cells, and then they get deflected by the organelles within the cell, and some of the lights can just deflect it off the cell membrane, and they basically the cells scatter light in different directions. So if we have some detectors around this area, we can collect those lights and measure them. So basically, the cells, each cell or any particle can deflect the light in different ways. But the lights which are scattered or deflected in 2 directions, one at the forward angle and the other one at the side angle, which is 90 degrees to the stream of the laser light, they are meaningful and they are actually proportional to the cell's property. So as the cell passes through, they meet the laser line and then they scatter the light. So these lights which are scattered and collected at the forward angle to the laser line, they are more proportional to the cell size. For example, a neutrophil, it can scatter the light in a greater signal, and which is bigger than the lymphocyte, for example. Whereas those lights which are scattered in the side scatter angle, then they are more proportional to the cell granules. And basically, for example, neutrophil has more granules, and its side scatter could be greater than the lymphocyte. So if we have some antibodies conjugated with some fluorochromes, which are detecting some antigen in the cell or within the cell or any part of the cell, then apart from these scatter signals which we collect, we can have some fluorescent signals as well. All right, and we can have a couple of colors. The reason that we call it because they are fluorescent light, so they're in different color. So we call it multicolor immunophenotyping. Oh, someone is. Oh my God, I think we did something. Oh, sorry guys, I think I'm back. Yes. Right, so now this brings us to the next part of the flow cytometer, which is the optics. So as we said, the cells are aligned and they pass through the laser line, and they are getting excited, and if they are conjugated with some antibodies, then they are excited and emit lights. So we need a very sophisticated system of optics which they can collect these lights, filter them, and deflect them to the appropriate detectors to be collected and detected. And once they are detected, they need to be analyzed by the computer. So basically, each fluorescent light now is going to be interpreted into electronic signals. So at this stage, we come to the next part, which is the data display or data processing. So we have three basic and more common data or system of displaying data in cytometry. One is histogram, the second one is dot plot, and the third one is quadrant. So let's see how they actually are generated and how they look like. So histogram, imagine the cell passing through and they just scatter signals. And in here in this graph, as you can see in the x-axis, we have fluorescence intensity, and in the y we have frequency of cell count. So as the cell pass through, I'll get one back. So as the cell pass through, they produce some signal, and the signal detected here. So the second cell passes, and then a second signal generated. So basically, what's the difference between these two dots or these two events is that this one has higher fluorescent intensity or bigger fluorescent intensity than this one. So if, for example, this is a forward scatter, if there's a forward scatter, that means that this cell has more fluorescent signal, more FSC intensity, which might be a monocyte, for example, and this one is perhaps a lymphocyte. The next cell come through again. We have a greater signal as well. So maybe this one is now neutrophil because it has a more fluorescent intensity in forward scatter, and this is a monocyte, for example, and this is a lymphocyte, and so on and so forth. So for example, this one, the fourth cell, is exactly the same signal for the fluorescent intensity of this cell. So we have two of these cells in that fluorescent channel, which means that there are, for example, two monocytes, for example. So as we said, the cytometry can analyze 100,000 cells per second and very quickly just start counting them, and eventually the software will smooth the histogram and produce a histogram. So now you know the meaning of the histogram, that's what's the difference of these events in here and the cell which is detected here. And eventually it gives a mean of this histogram. Okay. Now, for example, this is the forward scatter histogram. So we have lymphocyte peak here, monocyte peak here, and neutrophil. And we have, for example, solid scatter histogram, again, which shows the granularity of the cells, lymphocyte, monocyte, and neutrophil. And perhaps this is the common part with the auto-analyzer. They also give the scattergram. And if we want to show a two-dimensional plot, basically it means that we have, for example, a solid scatter on the x-axis, and we have forest scatter on the y-axis. And if we want to show these two parameters in one graph, we are basically generating a dot plot. And in this case, it is a lymphocyte population, monocyte population, and neutrophil population. And as you know, we can gate any of these population arms and do further analysis in other graphs. For example, we can gate this population and then show only this population and on the second histogram, for example. All right? So we can select any of this population. And counterplot, for example, also shows the frequency of the cells. For example, this here is in a scale of the cells, so that shows that we have more lymphocytes here than the neutrophil. And the quadrant is usually used when we wanted to have two colors in one tube, let's say. For example, in this case, we are analyzing CD8 versus CD4 lymphocytes. So we want to see how many of all T cells are actually T cytotoxic and how many of them are CD4 or T helper cells. So when we plot them like that, and we have, for example, PE conjugated antibody anti-CD4, and we have PITC anti-CD8, and we stain the cells, incubate the cells, and once we run it, so we can see for these two separate populations, basically. So in upper left, we have only cells which are positive for for CD4, and in lower right, we have only cells which are positive for CD8. And if there is any cell which is double positive, that would fall in this quadrant, upper right, which in this case only 2%, which could be because of non-specific binding of the antibody. So usually you know that we only have CD4 or CD8 T cells. There's no cell which is expressing both of these two antigens. And in this lower left corner, we only have the double negative cells. So that's the quadrant. And the way that the scientists analyze the data is, so once we want to generate, we need to generate a threshold for positive signals. So we run a sample as a negative control, which is usually stained with R-stop control, and then we set a threshold from wherever this histogram is ended. So from here to onward, wherever the signal come in, wherever the test is, then we can say that this percentage of the cells are positive. Okay, so we run the sample, and we can get, for example, CD4 percentages here. But there is a little bit tricky here, and that's why I said data analysis is very important as well, from sample processing, instrument setup. Even though your instrument might be good in terms of the quality control and meet all the quality control measurements, but still, when you set your multicolor experiments, you need to do all the adjusting, right? Say all of those steps are right, but now come to data processing and data analysis. So this is another reason that often we get different results from different labs. Right. So in that case, we have a negative signal here, and a positive signal is going to be like that. So the question is that still a negative signal? Because based on the threshold which we set up here, whatever signal is behind that should be negative. So based on that, it is negative. But it is actually a positive, because the difference is here. The cells which are positively here and fall in that part of the histogram, we call them bright, whereas the cells which are just shifted here, we call them dim positive. So these populations are still positive, but they are dim positive. So that's the place that most of the scientists make a mistake and then call this population as the negative. So there are a whole of different reasons that why there are shifted populations. That could be two shifted populations. That could be because of non-specific bindings. So this is the responsibility of that person who analyzes data to realize whether this is a true negative or whether it is a true positive cell. Is it because of non-specific binding? Is it because of the overlap between the adjacent signals, or any other reason? Right. So that brings us to some other challenges. Non-specific binding, as I said, this is a specific binding, this is a specific binding, but this antibody is just non-specifically binds to that, and even though it is non-specific binding, bind to this antigen, but it still has that fluorescent tagged antigen, and then can produce a signal. And the other reason that we may get some of these shifted population could be because of the overlapping signals. So because you remember the rainbow spectrum. So the cells, all these fluorescent lines, they are not clear cut at one edge, so they can overlap and produce some of the light can be collected by the adjacent signal. So we have to electronically remove this extra signal, which is detected by the adjacent light, by the adjacent channel. So there are a lot of adjustment in the system, and if the system is not adjusted properly, you may get some false result. And that might be actually, that shifted population, that might actually be true. As you know, as the cells are developing through the maturation process, they gain some of the antigens and they lose some of the other antigens. They lose more immature antigens and they gain some more mature antigens. That's why the expression, and that cell which is malignant might be in one of those stages which show the expression of that particular antigen not as clear and sharp as the mature cell, and we may see a shifted histogram. Right, so I tried to give you a very basic introduction to the system, how the system works, and what's the basics behind that. But it is definitely much more in depth about the flow cytometry. Any of those sections we can talk a lot, and there are lots of resources on the internet that you can read about. Some of the applications of the flow, just CD4 absolute counting for HIV patients, leukemia and lymphoma immunophenotyping, cell cycle, retic count, flow cross-match, for example, and a lot of other stuff like minimal residual disease, for example. And some of the applications research, we can look at the antigens at any part of the cell. We can look at even the cytokines. So basically, by flow cytometry, we can look at particles in size of cytokines, and we can look at particles in micrometers, like 1 or 2 microns. We can look at the plant cells more than 50 microns. So we have a broad range of particles in terms of size that we can look at, and we can analyze, we can tag them, we can fluorescent them, and we have a multicolor analysis. For example, we can tag the antigens which are phagocytosed by the cells, and we can identify those cells because they can detect within the cell as well. And we can do the cell cycle analysis. So basically, as you can see in here, we can G0, G1 cells, you can S phase and the G2 M phase. And if you put this beside that, you can see that any of these is actually one histogram itself. So basically, we have 3 histograms here, because which are different in terms of the fluorescent intensity. So if we stain the DNA with fluorescent dye, and the cells, those cells which are in G0, G1 has smaller fluorescent intensity, and those cells which are in G2 M phase, they have the greatest fluorescent intensity. And we can use it in cell cycle analysis, in fluid analysis of the tumors, and also if a cell is under apoptosis, which basically means So the DNA has been chopped up by the nucleases, and the cell has less nuclear material, then the signal would be much less than the G0, G1, and we can say, okay, this is the apoptotic phase, and they can gate them out and say how many percentage of the cells are under apoptosis. Right, and yes, this is a panel which these panels might be not the most updated one, but so in immunophenotyping we have panels for myeloid leukemias, a panel for lymphoid leukemias, we have panel for further down for blast identifications, because some of the leukemias are like M0, M1, or are unidentified, so we have to have a blast markers. We can stain the cytoplasm antigens like myeloperoxidase, for example. So this is just a very brief introduction to the panels for immunophenotyping. So I just try to very quickly go through the slides, and hopefully I could transfer the message that what the flow cytometry is. And now I'll just give you an example of the case study. So for example, a 60-year-old male complains with severe back pain, nausea, anorexia, and bilateral leg weakness. On examination, he was pale and tired and was in obvious pain, etc., etc. So the laboratory findings says hemoglobin 6, WBC 10, and normal chromic normocytic anemia, but there is no differential abnormality in terms of the WBC, but there's a proteinuria in the urine dipstick test. We look at the morphology, and we found some cells. Probably most of you are more professional than me, but you can see that these cells, because I know the answer, so I don't say anything. All right, so this is the image of the bone marrow, and now we have the flow cytometry. So we'll see how flow cytometry can help. So when we look at the scattergram, here is basically the more immature cells, which are read in red. So we can gate these cells out and then display these cells only in the next graph. Okay, and in the same tube, basically we have 1 antibody, 2 antibodies, 3, 4, 5 antibodies in this square. And you can see that, for example, CD19, they are negative for CD19. These cells are negative for CD19. They are positive for CD38 and 56. So this is the quadrant. So the cells which are in the upper right, they're all positive. And I would like to put this actually here. So that means all of them are positive. The whole population is positive. But the difference is these cells are probably a bit less in terms of the maturation process, whether it was in compared to those cells in here. But I believe that they all form the same cloning. And yeah, when we look at these cells, the expression of CD138 on these cells, we see that they are all positive for CD138, and they are plasma cells. So if I get back again to this slide, we can see yes, there are plasma cells. And case study 2, 65-year-old male, otherwise healthy, visit GP for noticing a slightly enlarged and axillary and cervical lymph nodes with no complaints. Otherwise, the football count in Australia we call it FBC. You guys call it CBC. The CBC showed WC 45,000, hemoglobin 13, and platelet 185. So far we have an enlarged axillary lymph nodes and no other symptoms. And we have a leukocytosis. The hemoglobin is just sounds normal. So this is the purple rod, basically, which, as you can see, there are lots of lymphoid cells. We can also go to the So yes, red blood cells sounds alright. So we have a lot of mature cells, lymphocytes. This is, what do you think, Ken, is this? And yeah, the majority of them are, looks like a mature lymphocyte. And if you go back to this, and this is the flow panel. Okay, and the flow panel, we have the scattergram. So basically, when we run the flow cytometry, when we get out any population of interest, then we can assign a color for each population. Then we can, it is called engaging, so we can look at the colors of each of these population, which we know what they are, for example. So if you have CD19 in this graph versus cytoscatter, so these are basically all B cells. So we get all the B cells here. And then we look at the expression of lambda and kappa, and you can see that 86% of them are only kappa. So this shows a monocularity of proliferations, right? So that means that B cells are a bit out of control. And here, what do we have here? We have CD5 versus CD20, and we can see that these cells which are going to be T cells, right? So these are the B cells positive for CD19, and these are going to lymphocytes which are negative for B cell marker. So we believe that there should be T cells, but we see that they are also expressing CD20. So these, about 75% of the cells are expressing CD20 and CD5 as well. And in order to differentiate the mantle cell lymphoma from CLL, we have a dual color of CD23 and FMC7. And if they are positive for CD23 and negative for FMC7, then that is more of a CLL. I think it's both our last slide. So this is Nightcliff in Darwin, Northern Australia. So sometimes I go in the afternoon and have a walk over there and take a photo. And I hope you all enjoy this. I can take some questions if you can text me. We can share all together. I don't know if people can text me or not. Okay, how can I invite questions? Mark is plenty.
Hey, excellent presentation, Alan.
Oh, thanks.
Yeah, I like the. Yeah, I think the organization of the presentation is excellent.
Oh, thank you.
Yeah,
I think Mark is now taking over, but I'm not sure he's not saying anything.
Okay. Mark, are you the technical guy? Yeah, I think, yeah, I'll have a chat with you later and give you some feedback on it. I think that overall it's a very, very good presentation, Alan. So thank you so much.
Thank
you can sign off now. Yeah.
Yeah, all right, cheers. Have a good time, everyone. Bye.
Okay, thank you. Bye-bye.
Bye. Bye.